Yeah, okay. Okay, thanks, Alex. Hi, everybody. I appreciate the opportunity to be here. So I'm Christine, and I'm currently a PhD student at the Center of Misfolding Diseases in the university. So I work under Professor Michele Ventruscolo, and well, most of what I do is working alongside the wonderful team at both the lab and in silico, where we work on identifying targets for aging-related diseases that are related to liquid-liquid phase separation mechanism. And so I hope that our work will make a meaningful contribution to the community.
So yeah, that's great. So see, you've got somebody in Cambridge locally who actually knows how to use PandaOmics and other tools, and is in aging for a while. So I think Christine started with us as an intern many years ago, and now she is at Cambridge in one of the top labs studying Alzheimer's. All right, so should we give it a few more minutes?
Yeah, we'll wait a couple minutes, and then yeah, then we can start.
Perfect. Let's wait for a couple more minutes. And yeah, Christine, if you like to tell everybody about your research and the wonderful fruits of this research that are likely to see the light in the next few couple years, that would be great.
Yeah, so recently, or in the last 1 year or so that I've been at the lab, we've been working on trying to select which disease might be the best model for us to study the mechanism of liquid phase separation. And so with the help of PandaOmics, we've been able to kind of prioritize which diseases have more proteins that are related to the mechanism that are also of importance. And so we've been able to identify Alzheimer's as one of the top ranking diseases. And using that, we've been able to further use the platform to kind of shortlist down about 15 targets, for which we are currently doing some target validation experiments. And hopefully, if all goes well, then our targets will be able to see the light of day.
Yeah, Alzheimer's is a big thing nowadays, right? Because we realized that many of the theories that were previously considered to be leading in Alzheimer's may not be as important as we thought they would be in the disease. So right now we definitely need more targets.
Okay, probably 1 more minute, and then yeah, then we can start. In the meantime, yeah, here's a cool, fun quiz. Wonder what everyone's perspective. How many years can drugs extend lifespan, in your opinion? Yeah, fill in the poll.
That's a good one. You should have more polls like that during the course of the presentation.
Or see how the results change during the time. Yeah.
Yeah, we'll re-poll this afterwards, after the talk, and then yeah, wonder how it'll change.
All right, guys. So whenever you feel ready, I can go, or you can start with the intros.
Yeah. Well, let's start. So good afternoon, everyone. It's a great honor and pleasure to host Alex today. So everyone, you may all have heard in Silicon Medicine making headlines a couple of weeks ago. So Sanofi, which is it's a pharmaceutical even bigger than AstraZeneca, signing a humongous deal with them. So yeah, everyone's excited to use the platform Insilico have built. Yeah, and if you don't know, Insilico are using AI and big omics data to try to find optimal targets to improve health and lifespan. But Alex will explain this much better than me. So Alex, the floor is yours.
Well, thank you for this generous introduction and also for the ability to present to the Cambridge Longevity Society. It's truly amazing that those societies are spurring all over the world and at top academic institutions. And I must say that when I entered the field, we didn't have any of that. And it's truly amazing how much and how little the world has progressed in aging and longevity. So I'll start sharing screen. I'll start sharing my screen. Give me a second. I hope you can see it. So just FYI, I'm on quarantine in Shanghai right now. So just in case I get disconnected, I'll try to reconnect immediately using a different network. I've had that problem before. And again, great pleasure and privilege to be presenting at the Cambridge University Longevity Society. And I'm going to talk about the possible applications of AI to identify dual-purpose therapeutics targeting aging and disease at the same time. And I'll kind of also give you a broader intro to the story. And I must say that when I was entering the field, I wish that somebody has had this kind of discussion with me and demoed how the industry looks like. So I had very broad, very rosy glasses entering the field in 2004. So when I switched the careers from information technology to biotechnology, so that was about 19 years ago. And I thought that the world is going to be moving much, much faster. So today I'm going to tell you why you should be very optimistic, but also cautiously pessimistic at the same time. And you really need to advance drug discovery before doing anything else. So I'm going to also present a little bit of a story of in silico medicine. So if you don't know the company, of course, it's great that you guys have picked up on the latest deal, but we are much bigger than that. And we've started our journey in 2014 in Baltimore originally. So we were born at the Emerging Technology Centers. It was at that time at the campus of the Johns Hopkins University. And then we spread globally and we're very Asia focused as well. So our Asian headquarters is in Hong Kong. Now the US headquarters are in New York. And we are also in Taipei, Shanghai, Suzhou, and recently opened in the Middle East and hired a lot of AI scientists who we can consider to be AI refugees from Eastern Europe, where we've got this terrible war between Russia and Ukraine, which we, of course, condemn in every way, shape, and form. But we also want to provide a home for some of the top AI scientists in the region, and we have an office in Abu Dhabi that is welcoming the top scientists from that region. One of the reasons why we are so Asia-focused is because the industry has changed pretty dramatically in the past 20 years. So now many experiments that you guys are learning how to do on the bench side can be done using contract research organizations. So CROs, I'm going to be using this word a lot today. And just like in information technology, where you saw major computer makers like Apple, Dell, etc., moving their manufacturing to China and completely outsourcing it, and even engineering to the Chinese CMOs, contract manufacturing organizations. In a very similar way, we see the transition of some of the research services being performed in Asia and also, of course, in many other areas of the world. But nowadays, you definitely need to know how to do those experiments. So please don't misunderstand. You need to learn and practice and know how to do the bench work. But when you scale up, you might want to outsource that expertise to somebody who is specializing in it. That's why we decided to be in this region. headquartered in Hong Kong, and then we oversee many contract research organizations in Asia. That was the logic for this global scale-up. At InSilico, we have the platform, which allows you to identify novel targets, generate novel chemistry with the desired properties, and also predict the outcomes of clinical trials. But the most value comes from the internal pipeline of therapeutic assets, where we're going after age-associated diseases, but also after cancer, immunology, oncology. So predominantly because that's what pharmaceutical companies want, and we need to ensure that we make money for the shareholders, not only focus on aging. I need to make all those financial statements, even though we're not a public company yet. No forward-looking statements, no investment advice. And just a few words about myself. So I started my career in information technology. I'm Latvian and Canadian by citizenship. And I did my first 2 degrees at Queen's University in Canada, computer science and management of information systems, and then worked in semiconductors for a while and learned the trade. So those same semiconductors are now powering deep neural networks. So it's graphics processing units, GPUs, that allow you to perform deep neural network computations at scale. And I worked for ATI Technologies for a while, made some money, and decided to, when I realized that I have, like a decade worth of savings, I decided to go into biotech and did my grad work at Johns Hopkins, and then followed on at MSU, worked for Sequenom for a while as a consultant, and also ran my 2 laboratories at the Pediatric Center for Oncology, Hematology, and Immunology, Bioinformatics and Regenerative Medicine. And then 2014, I started InSilico, and then I stick with the company till now, and it will probably be, I want to be buried in this t-shirt, because first of all, it doesn't degrade. It's polyester, and then it's making some really cool contributions to the society. But I'm very focused on longevity. So if you like to learn a little bit more about what I do and what I'm focusing on, you can go to longevitypledge.org. I decided not to leave the inheritance and focus all my life on extending healthy, productive lifespan for everybody on the planet, of course, myself as well, hopefully. And on that website, I outlined the steps that I think are needed to progress significantly in the field. And the first step is, of course, you need to transform drug discovery, because without it, we won't be able to move. If you're interested in practical applications of aging research, we've developed a course with a number of physicians and physician scientists. It's available for free at Longevity Degree. You've got 2 courses that are CME accredited, Longevity Medicine 101 and 201. And again, the industry will not move faster than the physicians do. You can do whatever you want in the lab, unless you've got the buy-in from the physicians, and now there are many of them. You will not be able to move. So that's why it's extremely important to educate the physicians, explain to them the recent breakthroughs in aging research and longevity, and also explain to them some of the concepts that their patients are going to be coming to them with after reading about some of those advances online. All of those courses are free. Another important tool that I recommend, learning resource, is a presentation by Michael Ringel, Dr. Michael Ringel, the managing partner at BCG, Boston Consulting Group, probably the best and the most influential consulting company on the planet. He gave a talk at our annual Aging Research for Drug Discovery meeting, so you can actually register and go there next year at the University of Copenhagen. I'll talk about it a little bit in the presentation. But in this talk, he talked about how aging research can be used as a platform for drug discovery for big pharmaceutical companies. And that lecture is golden. I mean, those slides are worth a lot of money, I know, because I know some governments that even paid for some of those slides. So or similar slides. So you really need to learn about the strategy for how to incorporate aging research into drug discovery and in the pharmaceutical industry in general. So it can be a very powerful tool for discovery of blockbuster therapeutics, potentially blockbuster therapeutics that will be treating age-associated diseases and also possibly addressing aging itself. That's what I'm going to talk about today. And also one side note is that if you completely eliminate cancer nowadays, so a lot of people are focused on cancer, a lot of money is going into cancer. If you are to completely eliminate cancer in the US, it's going to add about 2.3 years to life expectancy in the States. That's not my data. Many different studies projected similar results. At age 65, you are likely to gain 1.2 years to life expectancy. So it's not what you die of most of the time. And you die most of the time of cardiovascular disease, and that's where we can actually get quite a substantial longevity gain if we figure out how to prevent it and treat it. But in general, if you don't tackle aging, you are not going to get a very substantial, completely game-changing increase in life expectancy. You are not unlikely to double life expectancy if you even treat all three at the top. So we need to target aging pathways at their core. Many years ago, we started thinking about what is the most credible and sustainable way to research and commercialize longevity. Because one of the problems in the industry is that for many centuries, for millennia, humans were trying to tackle aging, and there were a lot of snake oil salesmen that made sure that aging research in general even sounds not credible. And anything that's dealing with longevity would be associated with vitamins or some kind of pills or snake oil, traditional Chinese medicine, and other things that most likely do not work at all, and complete fraud like homeopathy. So how do we figure out the best way to address aging research in a credible manner? And for that purpose, we've developed and organized the conference called Aging Research for Drug Discovery, originally in Basel in Switzerland. And now the University of Copenhagen is organizing it. Morten Scheibe-Knudsen, a very famous professor out of University of Copenhagen. You should follow him, and you should all consider coming to Copenhagen next year. It's a 5-day event where we actually discuss how to bring aging research into pharma, biotech, and medicine in general in the most credible way. So we also usually have big pharmaceutical companies presenting, and big pharma started warming up to aging research. So over the years, we've accumulated a substantial knowledge base on how to utilize aging research for drug discovery. And many of the companies presented, many business models were presented, and some companies were born with a presentation of this conference and died with the presentation of this conference. So if you look at companies like Restor Bio that were trying to spin out some assets, TOR inhibitors from Novartis, they successfully did that. They listed, they failed in phase 3, and the conference is going on. The first presentation they gave was at this conference in Switzerland. This year, we've heard the former CEO of Restor Bio presenting on another company, actually with a very similar business model. So trying again. And how AI can help solve aging. So how I got into the field, and we're helping. So you've probably all heard about the technology called deep generative reinforcement learning. Generative AI, you see it everywhere nowadays because Midjourney, DALL-E, Stable Diffusion, those models allow you to generate beautiful art. Before that, those are large transformer neural networks. Before that, we had GANs, generative adversarial networks. Now they can be combined, permutated, and there are many, many ways to utilize those systems to generate beautiful pictures and videos. But what we started using this technology for is first biological age prediction and monitoring. So I'm going to talk about it briefly. So you can predict biological age, but then you can also generate entire populations with age as a generation condition. And that's very valuable for target discovery, for data expansion, for anything. So imagine creating those beautiful virtual patients in time with a huge distribution of those patients across different age ranges. And surprisingly, you can use this technology for mental health, depression, age prediction, subjective age prediction, and also even identifying the best roadmaps for making people happier and younger psychologically as they age, because we actually also age psychologically. And a company called Deep Longevity, which was spun out of Insilico and sold to another company called Endurance Longevity, is doing exactly that. So I'm a pro bono consultant to this company, and you can follow my research papers on Google Scholar, specifically addressing this particular issue. I never thought that I would get into psychology, but now we started producing some very interesting results. But in biology, this technology can yield very substantial results in, again, biological age prediction for target discovery. So you can train deep neural networks on age, understand the basic biology of the process, retrain on diseases, and identify those dual purpose targets I'm going to talk about today. I can develop novel biomarkers. I can do molecular screening, molecular generation, so generate small molecules with the desired properties, can predict clinical trials. I can do clinical trials design more efficiently, expand patient populations, again, with age, and with different starting conditions. So you can use this technology across the board, and you can basically, it's the path towards the general pharmaceutical intelligence. So in 2014, GANs became a thing with the publications by Ian Goodfellow, at that time at the University of Montreal, and later Google, and then Apple, and now he's at DeepMind. So he invented this new technology, which allows two deep neural networks to compete. One is a generator, another one is a discriminator. One is generating meaningful noise in response to specific inputs, generation conditions. Another one is trying to see if the output of the generator is true or false. Together they compete, and once they're trained, they generate really precise objects, precise data, with the desired conditions, and these people in 2016 were generated using GANs completely. So nowadays it's impossible to make you to impress you with the Stable Diffusion and Midjourney allow you to create photorealistic images and even cartoon characters that have very beautiful desired properties with photorealistic features. But in 2016, 2017, that was really cool. And it also shows you that many features that can be used for generation of those images are retained and are extremely biologically relevant. So the most biologically important feature in anyone is age. It's more important than even sex, because age defines when you're going to die, pretty much, right? You can build a material model and predict the age of death of the person. And that feature is retained when you generate, and you can generate towards this feature. You can use it as a generation condition, and then use a predictive algorithm to identify, to predict this feature again. So this technology allows you to generate, again, massive numbers of people with a desired age, and with also different starting conditions, like, for example, a mutated gene, or lower gene expression of some of the genes, and you can propagate this this template into the future using age as a generation condition. Very powerful technology. In 2014, I got this idea that we can use deep neural networks to predict age, using many neurons on the input layer of the deep neural network and just one neuron on the output layer, and that we can use multiple data types that change in time. So transcriptomics, proteomics, methylation, imaging, videos, EEG, ECG, blood tests, even psychological surveys to predict age. And then once those neural networks are trained, you can utilize them to derive biological pathways, identify targets, derive causal graphs, and again, you can generate synthetic data if you turn it into a GAN. Or you can add one more neuron on the output disease and retrain the network using disease data as well. So this is a very powerful tool, and we've published a number of papers on it using this framework. My favorite data type is, of course, transcriptomics, proteomics, methylation, and also imaging data. So if you combine those four, you actually get quite a bit of insights into the cellular, into what's going on in the cells. So into phenotype, deep transcriptomic, proteomic methylation behavior. And you can do the same thing actually with live humans. So you can zoom in, zoom out. Given the enough data, you can train very sophisticated neural networks that allow you to understand the fundamental human biology. And at the end, I'm going to show you the robot that collects all four of those data types in one go. And you've probably heard about aging clocks. So the first aging clocks were published by Stephen Horvath, 2012, 2013. And he has a single author paper in genome biology. So that paper, of course, influenced me quite a bit and everybody else. And we started building deep neural network-based aging clocks starting 2014, 2016. We published for the first time. That was our deep hematological aging clock. We also got multiple granted patents on some of those clocks. Actually, those patents are important not because you can enforce the intellectual property that you created, but many people, it's now super simple to train those deep neural networks on different data types. People forget how to cite our papers. So it's actually a good idea to have a patent when there is a debate who published first, because a granted patent usually gives you priority. And we've published also deep transcriptomic clocks, microbiomic clocks. The first paper went on arXiv in 2018. And then we also started going into psychological age. So my first paper was in 2020. And I actually think that if you want to take an easy route, but also have substantial impact, go into psychology of aging, because that's one area of aging that you can easily modify. So not easily, but you can modify. And you can actually study the many algorithms that you would use for biological target identification and state modification using psychological data. So it's also a pretty cool thing to do. I'm going to switch gears a little bit from aging clocks, how to use AI to rapidly discover and develop dual-purpose therapeutics that can be targeting aging and disease. And to actually go after this goal, we established in silico medicine. So now we're spanning the entire globe. We have over 250 scientists globally, half AI and drug hunters. And we have over 200 full-time equivalents. Those are scientists within those contract research organizations that are, pipetting and synthesizing chemistry and doing biological research and creating the embodiment of your AI research. And we were recognized over the years by the many authorities. Top 100 AI companies in 2022, also 2018, top 10 in healthcare. And we started as an AI first company. So I actually originally thought that I would create a suite of tools for the pharmaceutical industry to use. And that's how we're going to be sustainable and we'll be able to go after grander goals. Well, it turns out that the software business in biotech is actually very small. So if you create the best of the softwares in the world, you would not be able to still demand substantial valuation for your company and grow. But if you use those tools, those software tools internally to generate drugs, and those drugs have very high chance of working, and you can tangibly demonstrate that, then you can create a lot of value. So I started as an AI first company. We started with a generative chemistry team, generative biology team. We are quite well known for generative chemistry. I think we were the first ones to publish in a peer-reviewed journal using GANs or small molecule chemistry. And we also did a lot of work on generative biology, but we did not publish as much. But once we realized that we can now generate pretty good target hypotheses and generate small molecules with desired properties, I met Dr. Ren, who was at that time with a company called Medicillin, a large CRO doing work for us. And before that, he was for 11 years at GSK in medicinal chemistry. And then before that, Harvard PhD, also medicinal chemistry. So you guys know, it's a good idea to go into medicinal chemistry because in my experience, medicinal chemists are the smartest people in pharma. They usually are the MMA fighters that understand biology and chemistry at the same time, and they can go into AI. So if you are planning a career in drug discovery, just make sure that you are good in medchem and also advanced cell biology. So Dr. Ren built a team of biologists, medicinal chemists, seroprioritization team, which allows you to manage those contract research organizations. And in many of those contract research organizations, we have our own people who are supervising those scientists. And it allows us to move very, very quickly. And the data that we are generating through our own programs is being fed back into the discovery engine, and we can very rapidly validate. And now that we've established that we can generate really high quality target hypotheses that work most of the time, we realize that it's a good idea to get some incremental data, and we're building. Actually, we're launching next month in December, hopefully the world's most advanced target discovery laboratory that can work in a fully automated manner and accelerate the cycle. But of course, you need to go through the cycle several times yourself with AI algorithms so that you can trust them. And I'm going to tell you a little bit of a story here. So this is my team in Asia, in Greater China. So this is me and Dr. Ren. As you can see, everybody is very committed to making other people live longer and prosper and better. And most of them have PhDs, and most of them come from big pharmaceutical companies before. We also raised over $400 million from these investors here. Many of them help us on the healthcare side. Many of them help us on AI. And some of them help us on governance. And of course, some of them provide very deep insights and oversight so that you don't turn into a company that is not as credible. So you need to ensure that there is governance oversight and a constant focus on creating value for the shareholders. So the better shareholders you get, the more oversight and the more help you get as well. And this is the most important slide for those of you who are not in drug discovery, and also for those of you who have those rosy glasses when talking about aging and longevity, and those interesting stories where we predict that in 20 years we're going to completely solve and cure aging. Well, this is how drugs are discovered. This paper, this slide comes from Stephen Paul's paper, How to Improve R&D Productivity, published in Nature Reviews Drug Discovery 2010. And in that paper, he looked only at this stage. So from target to hit all the way to submission to launch. So you already know the target you're going to go after. And this process takes you about 12 years on average. So here is a cycle time, and fails 90% of the time, more than 90% of the time. Actually, in the clinical stage, clinical phases, you get 10% success rate. And it cost at that time, so in 2010, it used to cost $1.8 billion. Now there are studies by Tufts and some other large industry and analytical research groups showing that it costs much more, so $2.6 billion per drug. Last year, the FDA approved only about 50 drugs. About 35 of them were small molecules, and in my opinion, about 4 to 9 of them were moderately innovative. So the entire world working for many years can deliver 9, well, basically, let's say 10 more or less innovative therapeutics globally after spending hundreds of billions of dollars. So currently, in my estimates, I think that globally we spend about $200 billion a year on research, and we get 10 drugs. Pretty crazy, right? But most important step that was overlooked in Steve Paul's paper is target discovery. And there I can gently put the probability of success at 1 to 5%. So 95 to 99 probability of failure. And it takes you about a decade to come up with a good hypothesis. Sometimes it may take you a eureka moment, and or you use PandaOmics. It might take you a day or a week, but usually it costs you a lot of money, and that's usually done in the universities like Cambridge, where scientists toil over the course of decades, come up with disease hypothesis, validate and test, and it's not just one PhD, it's usually many, many PhDs. So big pharmaceutical companies very rarely discover good targets and validate them all the way to humans. It's very, very rare. So I know only of a handful or less than a handful of stories where a target was discovered in a pharmaceutical company and they took it all the way to the clinic. Most of the time, this is done in academia or small biotechs. Then they take it to some level of validation. Then the pharmaceutical companies start looking at it, biotechnology companies start looking at it, and then the pharmaceutical companies pick it up at the late preclinical or phase 1, phase 2, phase 3 stage. And big pharmaceutical companies are there to commercialize. And I don't want to discount in any way the value of the internal research within those big pharmaceutical companies. Usually it's great, but it's just the level of productivity that is coming from big pharma is actually quite low. So today I'm going to talk about also a story where I went from disease hypothesis all the way to phase 1, closer to the end of phase 1 already. So hope to start phase 2 early next year. In a pretty substantial disease, potentially a blockbuster therapeutic, targeting aging and fibrosis at the same time. And when we started in silico, we looked at the different stages of drug discovery and looked at how long did they take, who performed those stages, those steps, and what are those steps and where AI could help. So as you can see, for target discovery and target validation, there are hundreds and hundreds of steps and hundreds of philosophies that we can take to identify a target and to validate it. And then when the time comes to prosecute the target with either small molecule chemistry or antibodies or some other biologics, you can actually do it within the pharmaceutical company or a small biotech, and then you start identifying hits, so the molecules that weakly bind to the target of interest. Then you optimize and optimize the molecules for safety, for bioavailability, for solubility, for many steps, for many properties, and you constantly test, right? So you need to, every time you do a cycle, you need to test in mice, you need to test in cells, and you test both safety and efficacy and many other properties. And then you perform IND enabling studies that allow you to start human clinical trials. So in silico, we actually, when we just started, we were of course focused more on target discovery, but we had this idea in mind that we want to go into chemistry and do the entire end-to-end. Many steps cannot be accelerated or improved by AI. So you actually need to do the in vivo PK/PD, you need to do in vivo efficacy, you need to do many disease models, because otherwise you won't be able to convince the regulators to actually allow you to start the human phase 1 or phase 2 study. So you need to perform many experiments before you go into humans. And what we are making as an objective function for ourselves, our main objective function is the probability of success. So we need to ensure that the target is right, and the molecule has all the desired properties that we needed to have, and we try to avoid making too many cycles where we synthesize, test, and have to sacrifice a lot of animals and go through many of those steps many times over. So by reducing the number of cycles and the molecules that you need to test and increasing the probability of success, you can actually reduce animal experimentation. You cannot completely avoid it. And I don't think you will in the next decade or so. And there are companies in our field, other companies that are also focused on target discovery, and many of them identify the target and then in-license the molecule or take it from a molecular library, so they don't do their own chemistry. They can maybe optimize a little bit, but usually they don't specialize in small molecule chemistry. And there are companies that are even in phase 2 at this point in time with an AI discovered target. And one of the companies like that is BioAge. So Kristen Courtney, who you definitely need to get on your one of your guest lectures. There are companies that actually don't think about target selection as deeply as we do, or as target hunters do. They pick the targets that are most commercially viable. So they look for targets that are likely to make a lot of money and also provide a reasonable challenge, so that they can explain that they have achieved something with AI that you couldn't do in the same time frame with just human intelligence. And there are companies, even in the UK, that have gone into phase 1 with an AI discovered therapeutic. But to my knowledge, we are the first one that managed to go both steps. So AI discovers a target and AI design molecule. And we managed to do this using the end-to-end approach. So we developed an end-to-end AI, which identifies targets for you. I'm going to talk about that in detail today. And then allows you to generate small molecules with the desired properties with as few cycles as possible, so of synthesis and test. And then we also predict the outcomes of clinical trials using the data from both of those engines and additional real-world data. And the main customer for this tool are actually hedge funds and banks that bet on small and medium type biotechs that have phase 2 assets. And we've launched the entire suite now for out-licensing. So you guys as students, you would not be able to get access currently to Chemistry 42 or InClinico, because those are the tools that are designed and developed for real heavyweights that allow you to deploy the tools on their premises. And Chemistry 42 is now used abundantly, ubiquitously in big pharma. So if somebody has, 1 or 2 brain cells and knows a little bit about AI, they usually deploy Chemistry 42 because it's a huge grand challenge in chemistry with many, many validated engines. And in my opinion, that's the, pretty much the only tool out there that allow you to do multi-parameter optimization at scale with huge validation. But something like that is available to pharma, and InClinico is available to the hedge funds and banks. And PandaOmics is used pretty extensively by academics globally. So every substantial key opinion leader group that is doing target discovery usually uses PandaOmics in one way or another. So there are only a few tools out there that allow you to identify targets. And now I'll switch a little bit to my favorite topic, and then we'll come back to PandaOmics and explain how we address target discovery in the context of aging. But nowadays, one of the main topics that is discussed at every conference, and sometimes you get even tired of it, is aging a disease. And my answer usually is that, what, should we even care? Who cares, right? So do you want to feel sick? Well, you feel sick right now. Is it a disease or not? I mean, we all have it. We're all aging. So what do we do about it, right? And from the regulatory perspective, the FDA probably doesn't care much if it's a disease or not, right? You won't be able to go through the FDA currently just with an indication aging anyway, because it requires a lot of capital and somebody needs to pay later on, for example, an insurance provider or national government to get that longevity therapeutic. And right now, the best way to go after aging is to look for dual purpose therapeutics where you can get some longevity dividend. Sorry for a spelling mistake here, need to fix it. So dual purpose therapeutics that give you the longevity dividend as a bonus. So you're going after disease, but then you can repurpose the drug into aging. And there are some examples that have not even been through human clinical trials for aging, but we've seen substantial life extending effect in mice and other organisms. And those are acarbose, metformin, rapamycin, acarbose plus rapamycin. So some of those drugs have the potential to significantly extend life. Maybe not significantly, but give you a few years, quality adjusted life years. However, you don't see acarbose and rapamycin being tested in human clinical trials for aging right now, even though there is a lot of evidence that they work. Why? Well, because they are actually not very novel. They've been out there for a very long time. I think metformin has been around for over 100 years now. Acarbose was one of the first diabetic treatments out there. It's reasonably safe, but it's very old and not novel, so you can get it for pennies in your local drugstore. And rapamycin is also not exactly new. Every pharmaceutical company has a TOR inhibitor, and some of them have what we call rapalogs. It's a sirolimus that is minutely modified for different purposes. Some of them claim that it's more selective towards a specific TORC protein, or maybe it's safer, or some basically has some other properties. But usually it's rapamycin with a tiny twist. Most of the time they did it for patent busting, to be able to get to the market with a really cool therapeutic that works and is a blockbuster. Many of them also targeted PI3K and TOR at the same time. PI3K TOR dual inhibitors. So rapamycin is a golden drug, which is used in oncology and immunology, and we are very confident that it works in many diseases, but it's not novel. So right now, if you are to actually go with rapamycin to aging and get it approved, well, you won't be able to make a lot of money because there is a generic version. So people will just buy it and use it off-label. It's very difficult to get IP protection. So if we are to do this all over again and to go for a new rapamycin and invent a new rapamycin, you want to do it from scratch and from the very beginning, try to implicate it on aging and many age-associated diseases at the same time, so that you can make a business case and take it into the market, and also try to use the aging biomarkers when doing a phase 2, phase 3 studies. So that will enable you to make an additional claim, but it will not significantly increase your cost of the study and require a separate study for aging. So, and plus it will be still novel and on patent, so you can still use it and make substantial amount of money in a disease. So how can AI help find targets that are common in aging and disease at the same time? And there are a bunch, right? So we know, again, TOR, we know PI3K, and we know many others that are implicated in aging and disease. So there is SERTs. And recently we've put out a paper in aging where we use PandaOmics to identify those dual purpose targets and map them onto hallmarks of aging. So actually, highly recommend looking at this paper. And in this paper, we took a large number of data sets from age-associated diseases, non-age-associated diseases, healthy people as well, and went through this PandaOmics workflow that I'm going to show you right now to identify promising targets that have various or varying levels of novelty and confidence And also we assessed drugability. So how much effort do you need to make in order to develop a drug for this target? And some are easier, some are more difficult. And then we mapped those targets onto hallmarks of aging by every hallmark of aging. By now, I hope that every one of you know hallmarks of aging. And we created this target wheel where the predicted targets are classified into confidence and drugability. Some of them are not novel. So you can actually find some drugs that might be already on the shelf or in human clinical trials for those targets. I can try to pursue an indication that is linked to cellular senescence, for example, extracellular matrix stiffness, etc. Try to test this hypothesis in that specific hallmark. Or you can go for medium novelty and high drugability, and medium novelty, medium drugability. Or you can go high novelty, medium drugability, and can create as a biotech a portfolio of those targets and have several shots on goal while still having aging in mind, right? And some of those targets are actually in more than one hallmarks. So please have a look at this paper. And I think for venture capitalists, that's a golden opportunity. We actually created a darts board out of this target wheel. So you can actually throw darts at some of those boards and see if you land on some interesting targets, and then you can read up on those targets. So now we want to have everyone use PandaOmics and also look for those dual target therapeutics. And we've started giving this platform as a demo originally, and then kind of in supervised mode, and then started giving demo access to a number of groups, and then also started subsidizing, giving grants to access this tool. And what really struck me was the ability to give this tool to high school students. So here you've got Andrea Olsen, 17, and Zachary Hapas, 16, present at our annual conference at the University of Copenhagen, and they presented their work on glioblastoma and aging at the same time, looking for those dual purpose targets using PandaOmics, and they got standing ovation. So they also had a poster. Hopefully they'll be able to publish. But this tool is so simple that a high school student can use it and actually get a pretty nice questions from the audience that actually knows the targets. You can see those target lists derived from PandaOmics on the screen. And now I'm going to show you how we used these tools for ALS. So ALS, amyotrophic lateral sclerosis, is an age-associated disease. So average age of onset is 55, 65. So you don't get it when you're a child, usually. Even though it's pretty rare, it has very substantial market size. So currently there are no standards of care out there, nothing that really works. So there is Riluzole and a few others, and there are of course many drugs in clinical trials, but currently there are no cures, and those drugs usually are symptomatic treatments. You don't get a substantial curative effect from those drugs. And what we try to do, many companies in our field in AI-powered drug discovery like to go after ALS and make big deals with the pharmaceutical companies right away before they even demonstrate the capabilities, because ALS is a very, again, promising disease for AI, because you can conduct some of the clinical studies pretty quickly, and the patients expire also very quickly, and it's a huge unmet medical need. So we went through the PandaOmics workflow where we used many omics scores to predict targets for ALS. We also increased confidence by looking at text-based scores. Again, target discovery is a balance of novelty versus confidence. So usually novelty comes from raw biological data, and confidence comes from text, right? So if somebody already implicated this target in a disease or in some very well understood biological process, you get more confidence in your target. Then we also have key opinion leader scores. We even assess the credibility of the scientist associated with the study and with the omics repository that we're going to be using for the study. And we also use commercial tractability, and we look at how similar targets or the same targets perform in biotech, because many biotechnology companies already tried many of those targets. They published annual reports, news, etc. And you can derive a lot of insights from those news. So you have to aggregate this data, process it, and see if any of the biotechs touched those targets before. And then we look at druggable classes, druggability filters, development level, tissue specificity, and many other features. So now I'm going to show you how to go after ALS with PandaOmics. It's an annotated demo. You can also find it on als.ai website. So this is how PandaOmics interface really looks. So you can go and search for ALS datasets. Here you will see the disease page. So you can see $2.6 billion worth of data in there. So we track biomedical grants. We link them to omics repositories. Here you can see the many omics repositories. The check marks shows that we actually did manual quality analysis, quality assessment. So somebody with a PhD or master's degree actually went into the data set and checked the quality. So you can select some of those data sets either from postmortem tissue or from iPSCs or some other source. You create data sets of control, case and control, and do some very simple differential expression analysis, differential gene expression analysis, also group genes into pathways, look at how those pathways are perturbed. We use our proprietary algorithm called iPanda. We published it in HComs in 2016, developed in 2013, 2014. So it's very validated, and we used it a lot before. So here you can even look at individual targets, individual pathways, look at how we construct those pathways and why this pathway is perturbed, either downregulated or upregulated norm to disease. You can also look at how they're structured, which elements contribute to the pathway perturbation, and also look at the relevant papers. So now we can create some of the comparisons and bring them all together. Create your own therapeutic area project. Let's say we call it amyotrophic lateral sclerosis. You start running the experiment. Takes you usually about an hour to run, so you can go have a cup of tea or go for lunch. But then you don't need to do it again. You've got multiple scores. Originally, you've got a weighted average of multiple scores, and you can see that the novelty is very low. We use more than 20 AI scores from, again, omics, text, KOL, etc. Each one catering to a different target discovery philosophy. So every column is a score, and usually it's either a validated or moderately validated approach to target discovery that we've accelerated. And again, when you first search for targets and create your first target list, you want to show low novelty targets. So when novelty is red, it means that the target is not novel. And you actually want the user to see it, because sometimes this user is the head of the therapeutic area in a big pharmaceutical company. And usually if they see the targets that they've never seen before, they lose interest very quickly. But if they've seen those targets before, they're like, okay, well, looks interesting. So your AI is producing something useful. And then we're like, okay, great. So now we're producing something useful for you. Everything summarized in one page. Again, you don't need to rerun it every time. It's already calculated for many different target hypothesis philosophies. And now let's look at different filters. So we can try to increase the druggability of those targets. So currently, as you can see, you've got a mix of druggable and undruggable targets. So now we've increased these druggability by small molecules, increased level of novelty to yellow. So now they're moderately novel. And you can see the list of some targets that you might not never heard of before. So like PTPRF and a few others, and some of them have never been implicated in ALS before. So now you can actually star some of those genes, and as promising targets, create your target lists and start cooking the targets. And once you are there, and once you've established some hypothesis for target discovery, you want to go to another part of PandaOmics, and that's the knowledge graph. So we just recently added some knowledge graph features that allow you to read papers more efficiently. So you can map those targets that you came up with into text-derived knowledge graphs. So we use transformer neural networks for that. And I'll just skip a little bit, right? So you can look at individual papers that contributed to the selection of each individual item into the knowledge graph. Like SOD1, for example, it's superoxide dismutase, right? So all of you know this target usually pops up in the literature for ALS all the time, right? Because it's oxidative stress target. And one of the hypotheses for ALS is ROS-driven, right? So reactive oxygen species-driven processes. And you can now map many of those ALS genes and select them and see how they relate to each other, relate to different biological processes, and also how they relate to different small molecules. So you can even look from literature which molecules work or were implicated in ALS and in this knowledge graph. You can loosen some conditions, you can add more nodes, you can add more edges, and formulate your hypothesis a little bit better. And if you're looking to publish, you can even screenshot it later on and publish or download the figure for publication. So this is also a useful feature of PandaOmics, which were recently introduced, but previously it was just part of the text-based target ID methodology. And we decided to run an experiment using PandaOmics, utilizing Answer ALS consortium data. So for that, we partnered with the wonderful consortium leaders, so Mary Chipkovitz, the head of neurology, chair of neurology at Harvard, and Jeffrey Rothstein. By the way, follow his research and look at his lectures. He's brilliant when it comes to target discovery, and Mary as well. But Jeffrey is just nominal knowledge base for targets. He runs, he was the co-founder of Answer ALS Consortium and the head of the Brain Institute at Hopkins. We also partnered with Bailu, very famous professor at Tsinghua University, probably the most famous in ALS in China, and Zhanke from Mayo Clinic at that time, who had a fly model for which resembles ALS in many ways. So when you're validating your targets, you're actually left with not that many choices unless you go into patients directly. You can try iPSC-derived neurons. You can try some other brain sources. Very, very difficult to obtain. You can try some animal models. So you can try to make a mouse model, use a mouse model that follows some ALS hypothesis, or you can use a fly. And those flies, genetically modified flies, are also pretty good validation exercises. So in this particular exercise, we took a lot of data from PandaOmics that's already pre-processed, gene expression data from ALS patient brain samples, Answer ALS data. Those are patient cells that were turned into baby cells and iPSCs and then matured and processed. and you have a lot of meta information available for those data sets. And we also used a lot of other public LS data. And we published a paper recently, so you can actually look at how we did it. In Frontiers of Aging Neuroscience, Dr. Frank Fong works in our Hong Kong Target Discovery Center, also runs our Hong Kong headquarters. And there, with this consortium, we published this paper where we predicted the number of targets and tested them in a fly model. So that's the G4C2 30 model. It's a fly eye model, which allows you to very rapidly test your target hypothesis and look at phenotypic modifications that a specific knockout of your target triggers. So you can see that for many of the targets that we predicted, we saw strong rescue and moderate rescue. So most of the targets that we predicted worked in an animal model, in a fly model, which is super cool. And we've predicted some targets and published them online, so you can actually look at them. And recently, we were invited to present in October. We were invited to present at a conference called Discovery on Target by Cambridge Health Tech. It's a wonderful conference. You should go there. And my head of AI platforms, Dr. Katrina Kamiya, suggested that, well, we show a small demo over the weekend. We do an experiment over the weekend where we generate small molecules for some of those targets. We select a few of those targets that we publish, and then generate generate small molecules with desired properties that might hit those targets. So we pick 3 targets based on biology from PandaOmics on this paper. We also predict the probability of success using in clinical. So maybe get it down to just 1 target, and then use chemistry 42 to generate small molecules with desired properties. So she comes up to me right before this conference. I'm like, okay, well, let's do this. It sounds cool. And she convinced our small molecule chemistry team that utilizes PandaOmics to the chemistry 42 very rapidly generate small molecules for some of the targets that were predicted were more likely to succeed in a phase 2 clinical trial in ALS. And we chose NR3C1 target, which had reasonable probability of success. And we have this tool called Chemistry 42, which has multiple generative algorithms. So think about a stable diffusion in every one of those squares. And some of them are genetic algorithms, some of them are GANs, some of them are transformer networks. There are over 40 models. In order for one model to be included in Chemistry 42, it needs to be validated. So we need to synthesize the molecules, test them, and see if they work. And then if the model got validated, gets a chance to get into Chemistry 42. And the way the model works is that you can give it either a ligand, already existing molecule for modification. So think of it as giving a face of Brad Pitt to Midjourney, and asking Midjourney to make this template, older, female, and with glasses and other properties. So we can do that, or we can take the target crystal structure, identify pockets of the binding pockets where the molecules may bind, annotate them a little bit, and give it to the generative system to generate small molecules with desired properties that might bind to the specific pocket. Once the generative algorithms get this input data, they start generating a lot of molecules. And as they generate, we have a lot of predictive algorithms that in 2D and 3D evaluate those molecules that are generated using generative system, and either reward or punish each individual model and also the ensemble for generating good or bad molecules, basically looking at how druggable, how drug-like those molecules are, and whether they are more likely to have certain properties. And then they also can be used, those predictive models can be used as a filter to filter out the best molecules that were generated by this huge symphony of generative systems. So you synthesize and test. So usually this is our kind of workflow. We identify the target, we give it chemistry 3.2, synthesize a few molecules, or maybe a couple cycles, get to a hit. So we generate and synthesize about 10, 15 compounds per cycle. Then we go for lead optimization. So that's when you start testing in animals. And every time, every cycle is actually a little bit longer. So you need to synthesize those molecules and test them in cells and animals. And then we get to a very good lead candidate and nominate preclinical candidate, right? So it means that we also test this candidate for efficacy in several models, and then we can nominate preclinical candidates. So that's usually the workflow. Usually it's not super long, but lead optimization may take a while because very often you want to have orally accessible molecules, oral administration. So you don't want to inject the patient with the drug, right? And if you want oral, it means that it needs to go through the gut and retain the properties, does not get degraded by all this gut machinery, gets into the blood, and then gets to the right tissue. So that's very difficult. You also need to ensure it's metabolically stable, it is bioavailable, and it's not toxic as well, it's not hitting something else. So this design cycle usually takes a little bit longer. And what we did is we took this glucocorticoid receptor, so NRPC1. We knew that there are some already promising hits and some ligands. So we took over the weekend one of those ligands and gave it to chemistry 32 together with the crystal, and asked chemistry 32 to make a better molecule, to optimize it. And since we were just running it over the weekend, when Petrina showed this, Dr. Kamia showed this results to medicinal chemists and biologists at the conference, they were like, oh, actually, it's those molecules that were generated and produced by chemistry 32 actually look great. And we would probably synthesize this one, this one, this one, this one. And it was pretty obvious why the system chose certain modifications for better properties of a possible drug for this target. So if we were, and then we of course did some 3D modeling, so looked at how those molecules may fit the pocket hypothesis. And then you also do a little bit of CADD and show a little bit more, get a little bit more confidence that this molecule, if you synthesize it, it's likely to work. So that presentation was very well received. But now imagine that we are not just stopping at drug design, and instead of just showing it at the conference, we go and synthesize and do a real therapeutic program. So in 2020, I got this, in 2019, I got this idea that why don't we utilize our entire software suite, just like what we've just showed you for ALS, but for some other disease that is age-associated, and we can go for novel target, novel molecule for a broad disease with no cure. At that time, it was a super grand challenge. So to my knowledge, nobody has done that before in a rapid fashion. Only a few pharmaceutical companies have done this internally using their massive resources. And we thought, well, we can utilize those contract research organizations in Asia and Europe to serve as a lab, and we will be just an operating system on top of them. Which disease are we going to go after? And I chose fibrosis originally, fibrosis in general, not only idiopathic pulmonary fibrosis. So I wanted to understand the general biology surrounding fibrosis, but we decided to purpose towards a specific indication. That's IPF, idiopathic pulmonary fibrosis. Like ALS, it's age related, so the average age of onset is also 55, 65. So within that age range, you don't see IPF in kids and young adults. And also, it's a huge market. So it's $3.5 billion for two drugs, nintedanib and pirfenidone, that do not solve the problem. So, and here is the workflow that we used for this therapeutic program. Remember, I told you that we developed those aging clocks that can be trained on transcriptomics, proteomics to predict age, and then we can retrain them on a disease. So we did exactly that here. So we used a deep neural net which predicts age and fibrosis, and then created large number of targets, large number of genes that might be associated with IPF and aging at the same time, with fibrosis and aging. Gave it to PandaOmics, prioritized many of those targets, did this novelty versus confidence assessment, prioritized 20 targets for validation. We could only test 5. And again, at that time, we were not very well funded. And I was doing it as a side project. Again, our main core reason why we raised from investors at that time was actually to develop software and take the software forward. But we decided to go samurai mode and decided to go after our own program. So we tested only 5. All 5 worked, and one target, I'm not disclosing this target yet, but this target so far worked in lung fibrosis, liver fibrosis, kidney fibrosis, skin fibrosis. In cells and mice, skin fibrosis, we have not nominated a preclinical candidate yet, but for kidney fibrosis, lung fibrosis, we nominated a preclinical candidate. Performed a lot of experiments for preclinical candidates. So for that, you need to do lung function experiments, for example, in mice, show that you can rescue the bleomycin-induced fibrosis in mice, preferably better than nintedanib. And we also showed synergetic effect with pirfenidone, even though the mechanism of action is completely different. You also have to demonstrate the efficacy in IPF donor-derived cells and many other studies. Then you do toxin mice. You do 14 days non-GLP tox, and you do ADME studies, safety tolerability studies, and then you nominate preclinical candidate. So we managed to do that in under 18 months. So again, in my opinion, that's the world record that we managed to set there, because it also for IPF costed us under $2.7 million. And we managed to do it very rapidly. Again, for you guys in the university, 2.7 million might seem like a lot of money, but once you do your own drug discovery, you start burning this 2.7 million very quickly. For if you are developing. And as you progress into human clinical trials, 2.7 million is actually very small. Then we completed multiple other experiments, so 28 tox in dogs, and additional toxicity and efficacy studies, and completed the IND enabling package, and decided to go into phase 0 human clinical trials. So this same time last year, I started the experiments in healthy human volunteers in Australia. So 8 healthy humans. By the way, if you don't know how phase 1 is run for a chronic disease, usually those are 23-year-old males who enroll in a study. You usually don't have females in a safety study for obvious reasons. And because you want to protect their reproductive ability, and you don't want to cause some unnecessary toxicity in the reproductive system. And why there are 23-year-olds is because those are usually university students that just need to get some money. And Usually those studies are conducted by contract research organizations. So we pay the CRO, give them the drug, they give us the phase 0 data. And once we realized and understood the distribution of this target and understood that microdosing does not really have any significant side effects, we decided to progress to phase 1. And now we are that close to completing phase 1. So fingers crossed. And of course, you get data all the time from the studies. So no bad news is terribly good news. And we are waiting to start the phase 2. And again, we did it in under 30 months. So I think that this is the first aging clock derived therapeutic, which is targeting aging and disease at the same time. But the validation is, of course, in phase 2, right? So once we pass the phase 2, hopefully, again, fingers crossed, then we will be able to claim major success, and then you can bury me in this T-shirt. And again, to my knowledge, that's the first drug where AI was used to discover a target, design novel chemistry, did some synthetic route planning, and we took it all the way into phase 1 after completing phase 0, with many experiments in between. And we're also working on skin fibrosis and several other fibrotic indications. But at Insilico, we are not only limited to fibrosis. It's only one program, or one target, several programs, 3 programs. We also are going after HIF-1 alpha, PhD2, for anemia and IBD. So there we design specific novel molecules with the desired properties, because there are actually some clinical stage compounds that are going after PhD2, but some of them failed in phase 3 in the US. So we wanted to ensure that for IBD, we have gut-restricted molecules. And Christian Fortney from BioAge, she also prioritized PhD2, which is very serendipitous. So we independently, completely different approaches, picked PhD2 for aging, and that kind of gives you a little bit more confidence in the target. And she in-licensed the small molecule from Taisho Pharmaceuticals in Japan, and is taking it to phase 2. So fingers crossed, and go Christine. Even though we're a little bit competitors, but I really wish them well. Great respect to BioAge. We also went after QPCTL target. It's a target within a CD47 pathway. And some companies actually prioritize it for Alzheimer's. So should have an age-associated angle. We prioritize it for immuno-oncology and reached the preclinical candidate stage with Fosun Pharma. So now it's co-development, and we got some really interesting upfront payment from them as well. And we own 50% of this target of this program, and hope to start human clinical trials as well. On the commercial side, we're of course very focused on synthetic lethality, so on cancer, and some of those targets are made for sale. So we want to get to a certain clinical stage where we will license it to a pharmaceutical company. So because we of course want to be commercially viable, and that's what I recommend everybody to do when they're starting a longevity biotech. Very often those longevity biotechs are just research for the purposes of research. You should have the shareholder in mind, and unless you establish a sustainable model, you should not expect late stage investors to continue bankrolling you in aging research. It's an easy kind of dream to have, to go after aging and think that people are going to be funding you all the time. Unless you figure out how to make commercially successful business model, you would not be able to sustain very long. And yeah, so we've managed to deliver multiple preclinical candidates since 2021, since Dr. Ran joined as a co-CEO. And now we're trying to accelerate this process with robotics. So in Suzhou in China, where many of those robots are made, we decided to build the fully automated AI-driven robotics lab. So to automate this PandaOmics process and ensure that we get more incremental data for target discovery. So imagine that you send the sample in, and in automated fashion, you get deep phenotyping, so deep imaging, deep transcriptomics, deep proteomics, methylation response. And we don't have humans in there. So as you know, in China, they have those AGVs everywhere, autonomous guided vehicles. They even bring you breakfast and lunch to your cubicle from downstairs in a fully automated fashion, navigating, competing for you with space in the elevator. So that's mainstream. You can cost down those robots. And in the lab, we have fully automated islands that allow you for automatic compound management, cell culture, high throughput screening, NGS, and also cell imaging and analysis. And in between those islands, we have those single arm, dual arm AGVs. So pretty standard equipment for cell culture. So you've all used those Hamilton liquid handlers in your labs. But we can do high performance, high throughput 3D, 2D culture. I can do iPSC, I can do organoids, I can do primary tissue derived from, mice and humans. And we also have very sophisticated high throughput screening platforms. We use those echo analyzers that allow you to perform high throughput experiments using echo. And we have an NGS system. We have single arm and dual arm robotics only island that can perform either very high throughput tasks, or they can even pipette like a human. You can teach them how to make some of the methods from Nature Methods protocols, right? And we have very standard NovaSeq 6000 that allows you to do RNA-seq and also methylation. And we have some other islands that I'm not going to talk about that we do not advertise yet. And we're currently piloting and doing field application testing. So once this lab goes into full operation, I'll be able to again do deep phenotyping, transcriptomics, proteomics, methylation, and then select the targets, prepare some of the compounds from libraries, and then test them on those cells. So incubate those cells with the compounds or organoids, and then do again deep phenotyping and transcriptional proteomic methylation response, which is pretty cool. And that will allow us to do aging experiments at scale as well, hopefully, again, as a side product of our drug discovery business. So that's it for today for those dual purpose therapeutics. And I would love to open the floor for questions.
Okay, that's pretty crazy. That's a lot to unpack. And that's a lot of really cool stuff that you've shown. At the start of the talk, while we were waiting, I asked this question: how many years can drugs extend lifespan? I wonder if we relaunch this, what sort of numbers we can get. But yeah, do you have any sort of questions from the audience? We do have a couple. Okay, first I'll ask Milan. Feel free to ask the question yourself. Just tell me. But first we have Milan, who had to leave mid talk. He's asking about so we all have sort of genetic diversity, and he's wondering, in your sort of PandaOmics platform, how important is genetics in making these predictions? And yeah, that's
of course. So genetics is of course extremely important, right? So we all are made from DNA at the beginning. But when discovering targets, I actually have not seen many genomically many targets that were discovered using genomics data, right? So there are a few, like for example, PCSK9 and maybe a couple others, blockbusters that came out of genomic data. And also, there are a lot of mutations that were picked up, specifically relating to some cancers or some other diseases. But usually, you cannot find. You should not use purely genomic data for target discovery. It's not the golden kind of. It's not the golden pot of target discovery. For target discovery, it's a good idea to use genomic data for target validation and also for patient stratification. So, but there are some target hunters out there, and heads of the therapeutic area that will not pass the target into early preclinical studies or late preclinical studies if it's not genomically validated. So genomic validation, we do have a tool in PandaOmics, and two of the scores are genomically driven. So if you have this fetish for genomic validation and genomically validated targets, you will be able to get that through PandaOmics. Is it a good data type for target discovery? No. I think that the best data type for target discovery would be proteomics plus methylation plus transcriptomics. So those three are, when you have a good combo, it's a very good data set for target discovery, especially longitudinal.
Yeah, right. I guess following on from that, because he's asked a secondary question, can PandaOmics predict, or on a population level? So, 60% of people will respond well to these targets, or yeah, I'm wondering.
Very good question. Yeah. So you basically and we use this this question in target discovery and also in prediction of clinical trials outcomes. For doing target discovery, you actually want to first come up with a data set. I just showed you an example, right? So a data set of people with similar features when it comes to disease, right? So you try to look at the patients that are as close to each other as possible for original target discovery, right? And then you create your target lists using those people. Then you can narrow down and see if the targets that were important for that patient subpopulation are also very, very important in the top, let's say 3-5 in individual patients, right? And that's how you can assess target heterogeneity. So if the target heterogeneity is huge, right, and you've got major variability in those top 10 predicted targets from patient to patient, it's actually not great, right? And also that is something that will influence the outcome of phase 2 study. The more heterogeneous the target is, the more likely the trial to fail. And PandaOmics allows you to do that. You can even use a data set of 3 patients to identify a bunch of targets out of those 3. And my recommendation is to start with as many as possible and then try to narrow down and also come up with a reasonable biomarker hypothesis to be able to narrow down the patient population when you're doing your phase 2.
Yeah, I guess everyone here is pretty excited to, or they would. There were a lot of us are academic researchers. Abjit is wondering, can we use PandaOmics or as academic institutions? I know you mentioned. So what's the process? How do we? We're excited to try these tools.
Yeah, you can request a demo online. And get trial access, and afterward you might want to get your institution to get your license. And some institutions, they already have a license. but it's better to try it yourself. And also, if your project is very interesting and you are not going to give us any data, so we are not interested in getting your data, by the way, as the last thing we want to get. We can also have a collaboration where we would go after a specific target hypothesis, methodology that you might have, right? So if you have a good idea on how to discover targets in a new way, we can collaborate and see if your target generation methodology can be compatible with the other scores in PandaOmics. And then if it's really good and if it's validated, we might consider kind of extending it and even integrating into PandaOmics in later versions. That's how many of those scores came to be, right? So through major collaborations with pharma or academics, etc. So we're trying to constantly improve the tool. And if that collaboration can help improve the tool and even usability of the tool, we are open to collaborations. But you can request a demo online, and we have a team of application scientists who are looking at the projects that people are proposing, and if you can propose a project as well.
Yeah, I suppose a lot of researchers or a lot of academics would be grateful to find these targets, and then it becomes a matter of validation. And then they would, I'm sure they would happily, or some of them would happily share back the validation data with InSilico. Okay, yeah, Max, you've been waiting patiently a long time to ask a question. Please go ahead. You're muted, by the way. Okay.
Thanks. Great talk, Alex. And there's just so much that you covered, and we never kind of seem to want the talk to end. And there's just, I'm sure you have a lot more to talk about. So thanks again for talking to the society. I find your approach of dual purpose drug discovery really interesting, because you touch on business model and how to make things sustainable. I have kind of maybe two questions related. First, that when you find a drug that ultimately, let's say, get approved by the FDA for a particular condition that is not aging, then how would you imagine people will be able to use that drugs for aging, and how it will be prescribed? And do you think ultimately FDA will classify this as a disease in the future, and maybe ending changing the endpoint, so how they kind of approve drugs like in this area?
So that's let's wait and see how our program 1 goes, right? So before you go and start purposing anything into aging, you need to ensure that it's very safe and beneficial, right? So if you see major improvements in certain biomarkers, then you might want to start looking at off-label use, right? And of course, in our case, we want to go after the broadest indications possible, right? So first, it's going to be, well, IPF. It's not huge indication, right? Even though it's pretty profitable. But then there are many diseases that are driven by fibrosis and that have fibrotic origin. Like, for example, kidney fibrosis. If you think about it, it's like 15% of the global population, right? So if you have this number of people, patients using the drug, you will be able to see the effects in aging and longevity. And once when you're conducting the phase 2, phase 3 clinical trial, you can actually use some of the aging biomarkers to come up with a hypothesis that it's implicated in aging. And you're possibly even reversing it, right, at some level. So when you're at that stage, and when you are, again, commercially successful with a drug, but you still have the patent life and you own this small molecule, you could do a lot on aging. in my opinion. It's too early to tell what the FDA is going to do, right? And how it's going to react. But we see right now, for example, with rapamycin, there are rapamycin clinics out there that are prescribing rapamycin to healthy people, right? And I recently did a survey of the academics with H-index of 30 plus, and many of them are taking rapamycin. So of course, there is no exact protocol on how to do it, right? But I think that once we get some significant evidence from large clinical trials for a specific disease by a company that actually really cares about aging, right? So like us, for example. So we are not going to cut the corners. And we're going to look at all the biomarkers, right, that are there. And some of the biomarkers that can be used for aging research are necessary to conduct the phase 2, phase 3 clinical study anyway. So you would have a biomarker augmented study. And then you could repurpose, right, hopefully, with much more evidence than what we have from rapamycin right now. Because rapamycin is used for cancer and organ transplantation. Those are not the two indications where you can gather a lot of populational metadata implicating the drug in longevity, right? So because those people usually have a big problem, right?
Yeah, that's sorry, did you want to say something, Max?
So that answered the first question. So the other question I would like to ask, so I guess, this, you're doing it in a proper pharma traditional way, getting FDA approval. So do you think there are other opportunities for kind of innovative business models, or like people who are, let's say, focusing on maybe herbal medicine, traditional medicine, so things like nutrition, supplements and things like that, where AI or your platform can actually be applied to find certain things that are, let's say, natural products or people have been taking for thousands of years already traditionally. Do you think those may potentially be useful also?
So look, if people were taking natural products for thousands of years, where are those 120-year-olds, right? I wonder whether they are, right? If traditional medicine works. But to answer your question, yes, we tried that before, and you can actually look at some of our early papers on geroprotector discovery. We have a series of papers where we developed this tool called Geroscope that looks at transcriptional response of many cells to a very large number of compounds. And then we use this data to identify molecules that may have some geroprotective this data to match some of those molecules to natural products, to like N-acetylcysteine, EGCG, epigalactine, epigalactine galat, and what else? Vitiferin A, and a few others. And then we gave this data to Life Extension. LEF, you probably know this guys very well, one of the large supplement makers, and they made a supplement. So on the bottle, it actually says that based on research from in silico medicine. And N-acetylcysteine, I think that it's a very well-known compound that worked in mice and other organisms in terms of life extension. And we made a combo, but very often, since it's not a pharmaceutical, you're not conducting a clinical study on something like that, right? And it's very difficult to get a blood test, and people usually are in the wild, right? So unless we see somebody who is deeply committed to this project or product living to 120, we won't know if it worked or not, right? And again, there is very difficult to get the data from those real world nutraceutical enthusiasts. So my recommendation is not to go that route, because you're not going to make a huge impact. You might make some money. And with my lab, I can actually test some of those compounds for different drug effects, right? So even TCM extracts, you can try and use in my lab to see if you get certain phenotypic, transcriptional, and methylation response, right? But that's where it ends. So after that, you need to to drill down into specific molecules and try to go after a specific disease. So I think that going pharmaceutical route is the most credible way to operate today. If you want to make a bigger impact than pharmaceuticals, you should go regenerative medicine. So that's where the real potential is, in my opinion, for anything longevity.
Right, right, absolutely. Thanks very
We're
much.
going to see the results of the survey. I'm very keen on seeing how much do people think that the pharmaceuticals can extend lifespan. It's actually an interesting exercise. But my personal belief is that, yeah, well, you can probably get 20 years. Maybe you can get more than 20. Maybe you can get 25 if you ultra optimize, if you are ultra healthy, and plus you're on all the drugs in the right time. But I think that's where it ends. So you shouldn't expect miracles.
Yeah.
But with the regenerative medicine, you might.
It's sort of a competition between, I guess, a lot of people are subscribed to the DNA damage sort of hypothesis of aging. It's a competition between how much time you have on from DNA mutations and so on, and versus also there's this new concept, hyperfunction theory. I don't know if you've heard of it, Alex, or sort of like overdevelopment theory, where me personally, I think in silico, I'm personally really bullish on in silico, because hyperfunction theory is this idea of like unguided development, and past reproductive age we're no longer optimized towards, you know, development. For example, we may have pathways like inflammation that are chronically switched on that don't need to be switched on, and sort of all these different genetic pathways can be sort of targeted and modulated to maintain homeostasis as long as possible. But yeah, apologies.
Yeah, well, theories are of course good, right? And you've probably heard about antagonistic pleiotropy theory, right? So some genes that you need for development later on are pretty harmful later in life, right? So it's a very well established theory, but of course there are probably 1,000 theories out there. So every aging scientist who is pretty established and who I know and who presents at ARD, they usually have their own theory, and they came up with a term or two. But all of that is valuable. It's extremely important to understand what aging is, how it transpires. But right now, I don't think you could dramatically intervene in aging with drugs. So if you were to do a thought experiment, to conduct a thought experiment, and again, it's a thought experiment, it's not a motivation to go into that area. And I just published a Forbes article on that. So imagine that you are constrained to all the current technologies, right? And that's it. So stop everything you're doing, right? And let's optimize what we have. So if you were to try to significantly extend your lifespan right now, and you probably can live as long as your brain can, right? So the brain is the ultimate organ, which once dead, that's it, right? And you want it to function for as long as possible, but everything else is actually already replaceable, right? So transplantation has progressed pretty dramatically. So if you were to grow a, let's say, unconscious clone, and most likely with some of the with some of the technologies that are available even today, you could probably grow unconscious clones by disabling the higher functions, right? Then you could probably transplant a bunch of organs, including your muscle and other tissues, onto yourself, right? And even go with something as crazy as brain transplant. Again, it's just as a thought experiment. It's not a recommendation. It's not what I'm doing. But imagine how much that would extend your lifespan, right? For a second. Because you can also replace your face, right? So yes, you're going to be left with the old brain, but everything else is going to be new. And it's a pretty dramatic thing, but it's very compatible because it's your own clone. And maybe you can, the only objective of that clone would be to exercise and diet all the time. So if you do it early in life, well, you've heard about parabiosis, so I'm pretty sure your brain would benefit from that. And that could add additional maybe 50, 60 years, right? We don't know, maybe more. And that's something that's already available. But I think that you go into drug discovery to, of course, also fight disease, right? And it's the most credible thing to do. So if you are to go and do brain transplantation right now, I'm pretty sure that's not something people will perceive as very credible.
Yeah, no, that's interesting because there's a researcher in the US who sort of has this idea, this pizza model idea, where you replace small like chunks of your brain at a time. And so that's, I don't know, one.
Yeah, I wrote about him in he's actually a very credible scientist.
Yeah, no, it's interesting.
From Albert Einstein, yeah.
And interestingly, our current audience, 83% of people say they would transplant their heads into a younger clone of themselves. So that's anyway.
You would be surprised that a Twitter poll, 790 people said that they would.
Out of what percentage is that though?
So it's more than 70%.
Right,
so
okay.
75% said that they would. Sorry, 70% said that they would, 15% said that it's maybe, and 15% said that they would not.
Right.
So that was extremely surprising. That's why I wrote an article. Yeah.
Yeah. No, Alex, thank you so much for your time. I know we're running really late, and are you happy to stay? I don't know, 5 more minutes for some more?
Well, yeah, sure. I'm in quarantine. I can.
Okay. Yeah. No, we have a ton more questions. We have a couple more questions. Just maybe quick fire your thoughts on them. Michele Frison, do you want? He's wondering about post-translational modifications, and I guess from an omics level perspective, it's much more difficult to sort of get a lot of data for post-translational modifications. Yeah, Michele, feel free to ask your question if you want. But yeah, I'm wondering your.
So I can expand on that. Of course, it's very difficult to incorporate that into target discovery. However, you are likely to see the effects of post-translational modifications in both expression and proteomic transcriptomic data and methylation as well. So using those data types, you could potentially narrow down to the troublesome proteins that went through post-translational modifications, and also anything that happens at the RNA level, you would probably be able to see that through this data.
Yeah. And yeah,
so that's why I think that deep phenotyping, transcriptomic, proteomic, methylation data, those are extremely important. So, and of course, again, deep phenotyping, I'm talking about high-res imaging.
Yeah. Oh, Michele, did you turned on your camera? You wanted to say a few things, or?
Thank you. No, just thank you for the talk. It was a great talk. I'm just wondering, basically, if you think that there's any technologies downstream in the future or right now that will really like what's happened for sequencing, allow for the omic analysis of many post-translational modification at the same time, if that's something that's interesting at all for somebody working in your field. Just about
well, I think that of course is very, very interesting, but right now you've got already many tools to do that, like mass spec, for example, right? Many other imaging techniques, right? So imaging is becoming better and better. I'm pretty sure that with time we will, and with, again, substantial academic resources, we will be able to look deeper into post-translational medications, and also anything that happens even on the like protein damage, right? So very often we don't understand that too, and what how proteins are damaged. I would love to get deeper insights. Unfortunately, with my lab, for example, I'm trying to go after the lowest hanging fruits. And again, PTMs is not the lowest hanging fruit.
Understandably. Thank you. Thank you.
Okay. Yeah, so we have a couple more questions. So Nell is wondering your opinion on well, so, yeah, she says rapamycin and metformin both modulate mitochondrial pathways. How big an impact do you think mitochondria are in aging? Or yeah, just your general thoughts on that.
I think it's a huge impact. Mitochondria plays a huge role, but it's not the only factor. So aging is such a multifactorial process that I wouldn't be able to even weigh a certain organoid or a specific process in terms of how much it contributes to aging. But of course, yeah, metformin definitely implicated in mitochondrial function, right? So multiple papers there. So rapamycin indirectly as well, and definitely there is something there with mitochondria in terms of the recent animal experiments. And as we know, there are multiple. Well, there is a couple handfuls of proteins that are being coded in the mitochondria, right? So we also might want to look into that, and there are many groups that are looking at mitochondria in the context of aging, but we are not exactly looking at mitochondrial genes or specific mitochondria targeting drugs for anything that is drug discovery at this point in time. I looked at mitochondria a lot when I was consulting for Colby Pharmaceuticals that were making those mitochondrially targeted drugs, mitotemples, and derivatives of mitochondrially targeted quinones, etc. Right now, there is MitoQ, for example, out there. It's the quinol linked to triphenylphosphonium, and I don't think it worked in aging or anywhere. So now they're selling it as a supplement. And yeah, of course, mitochondria plays a huge role. I'm not exactly very bullish on mitochondria right now. Possibly huge role in Alzheimer's, right? But
yeah,
I guess not what I'm focused on.
The diseases like maybe Parkinson's, or these sort of very energy metabolically demanding organs, possibly. But yeah, I know you showed it at the beginning of your talk, sort of the cancer. If cancer is completely or solved, it would add 2.3 years. The biggest one was heart disease. So 10.9 years or something in that slide. So I don't know how much mitochondria. I don't know if in like cardiomyopathies or these.
Again, I wouldn't give a weight, right? So I made my choice. My bet was on fibrosis. And still is on fibrosis. So I think that if you know how to regulate this process first, it will give you a huge toolkit to go after many diseases, and also it's an enabling technology to go into regenerative medicine, right? If you are not going to go for whole body transplant. Yeah, so fibrosis it's a big thing. I think that is probably the biggest thing.
I guess that touches on Ijo Yu's question, because he was wondering about your thoughts on mitophagy. I guess we've sort of spoken on that. Unless Ijo, you want to add a few more things, then just let us know.
Sorry, Brendan and Alex. Super great talk. I think you've actually answered my questions in the previous questions. But yeah, great talks. Thanks for.
Yeah, but not to discourage anybody. I mean, mitophagy is an extremely important process, and
yeah, I think quite a few of us
drugs that regulate it. Yeah.
In the audience, actually, a few of us work on mitochondria, so I don't want to break the hearts of anyone.
No. Again, I'm very, very bullish on that. I just don't think that I can work on that right now. So unless there is a sponsored program with big pharma where they're going after mitochondrial targets, I would not be able to go into mitochondrial targets myself. And so sorry guys, I have to be always mindful of my own kind of kitchen, right? So if you are cooking in the kitchen, and if you are cooking Italian, and somebody is saying, look, you know what, sushi, what do you think about sushi? Like, great, but I cannot switch well, actually, from Italian cuisine. I would say, okay, well, Chinese, right? So it's difficult to switch from Italian to Chinese.
Yeah, no, I wonder if AI can put a coefficient on different sort of organelles. So maybe these organelles are sort of more limiting. But anyway, yeah, no, Alex, it's an absolute.
That's actually that would be a really cool experiment to conduct at the system level, because I think that in terms of cellular senescence, there has been many nice papers showing how different organelles behave, but in terms of systems level, I don't think there has been a good experiment. So something that you guys should do.
Yeah, no, me personally, I'm pretty subscribed to the DNA damage theory. I agree with sort of, it's like, what, let's say 50%. So I mean, if you measure how many mutations there are, by the time you're 80, it's something like, in your genome, you have like 4,000 mutations. Or this is a paper by Alex Kagan, 2022. And so it's interesting, 4,000 mutations by the across your whole genome by the time you're 80. So our genes, we can sort of withstand sort of 20% of our genes having one mutation on average. But yeah, no, for mitochondria, also just across different animals, if you look at mitochondrial mutations, it's sort of much weaker correlation with lifespan, or it's slightly weaker correlation with lifespan compared to like nuclear somatic mutations. Anyway, so, yeah, just trying to see if we can quantify, how important are mitochondria in aging. Anyway, but yeah, no, Alex.
It will come to
Sorry? Oh, so we lost you a bit.
Aging research. That's all of you should come, and that's really.
Okay, I will mention the comments that Alex is sort of dropping out a bit.
Right.
Sorry, you're dropping out in connection.
Yeah, I guess.
Okay, because I can hear and see you very well.
Yeah,
but
sorry.
I'm not sure about.
Yeah, unfortunately, like 85% of what you were saying sort of dropped out, and we couldn't quite get it. But yeah, we see the link.
Sorry, just the last 15 seconds before you drop off. Can you repeat that, Alex?
Yeah, I'm just saying that it's a great idea to come to the 10th ARDD.
Absolutely.
And at the University of Copenhagen, and spend 5 days with the thought leaders discussing those questions. Because I guess in my case, I'm more of a drug hunter now, right? And all I think about are targets, and drugs. And when you want to have a broader perspective, it's also great to have a bunch of theoreticians, so people who are in theory, and also a bunch of medical doctors. Because if you're subscribing to, let's say, DNA damage, DNA repair theory, if you talk to an MD and see the physiological representations of aging, you will see that it's, again, such a multifactorial process that in many cases, DNA damage, DNA repair doesn't have anything to do with it, right? Like extracellular matrix or cellular recycling, again, fibrosis. Once you see those biological processes driving diseases and also loss of function, you can try to link it back to specific theories. And we've, by the way, tried doing this. So we had this resource called Aging Chart. That's now offline, but a lot of thought went into that. Try to create kind of a pathway level Wikipedia of aging, and also at different levels stemming from molecular processes. And I think that DNA damage is, of course, very, very important contributor to aging, and we know that, but it's just such a multifactorial process that it's I wouldn't pinpoint it to a specific theory.
So if I may add some comments, really great insight also and approach. I guess you mentioned lower hanging fruits, and I think the approach where you take and kind of the business model is really fascinating for those of us who are more interested in startups and entrepreneurship. If you remember, I just finished my EMBA in Cambridge, and so I kind of think more about the business model side. And obviously, we have lots of PhD students here who are at the forefront of their research, and we need those pioneers. So definitely, I think picking fibrosis is really and all the best with that, and we all need more drugs related to aging diseases. And I just want to say something. So we are very lucky to have Alex in person next year, and I think it would be a bit, hopefully the topic would be a bit more entrepreneurial also. I think people would love to hear about your story, how you fundraise, and how you kind of approach startup process. So that would be a great opportunity for everyone interested in potentially commercializing their research and doing something in longevity in terms of startup. And what better place to do it than Cambridge? So one of the best places yeah, we will look forward and thanks for this talk, and look forward to your in-person talk next year. And it will be quite entrepreneurial. We'll try to make it, we'll try to work with other entrepreneurial societies and colleges to do that. So Brandon, over to you to
yeah, I agree. Alex, final remarks. Alex, I just want to say, I love that dartboard. I would love to buy that dartboard with the targets of aging, and I think we might. It'd be fun if we make a social event. On sort of that sort of thing. But yeah, no, Alex, it was an absolute pleasure to have you for the talk, and thank you so much for staying. We only expected in 1 hour. We're very happy to. It's also a good thing that this is recorded, so we can sort of distribute this whole discussion online. But yes,
well, it's Saturday night, so nothing better to do except for writing papers. But yeah, anyway, guys, so great pleasure, and also, I kind of dedicated everything I have to longevity research. So also send me the society's bank details. I'll send you a few £1,000 for to have a party.
Oh no, that would be absolutely amazing. Thanks, Alex. Yeah, no. Thank you, everyone, and we'll see you at the next
Cheers,
event.
guys.
But yeah,
Yeah.
bye, guys.
Take care in the
See ya.
meantime.
Bye-bye.
Take care. Bye.